For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
All right,
Hey,
it looks
everybody.
like we are live. Hey, Ramat and I are extremely excited for this interview. I got to say, it is an absolute privilege to interview Dr. Rob Stadler today on this very important topic. That being said, Rob is no stranger to this channel. You would have seen him in an interview with John Maddox, as well as one of my personal favorite streams on this channel, Educating Professor Dave. I do want to pull that up on the screen for everybody. Anybody who has not yet seen it, please do check it out. It's definitely a must watch. Share around. We definitely want to get it out there for as many people as possible to see. And also check out Dr. Stadler's interview with John Maddox. Definitely worth your time. So anyways, let's get right into this interview though. First, I want to thank Dr. Stadler for being here and giving us his time for this. Whether you are new to this topic or you have been studying these issues for years, you are sure to learn something today. I'm going to hand it over to Ramat to introduce our guest. Ramat, the floor is yours.
Awesome. Yeah, thank you again, Dr. Stadler, for coming. And this is for everybody who doesn't know. Rob has been the author of two must-read books. One is titled The Scientific Approach to Evolution, What They Don't Teach You in Biology, and The Stairway to Life. Those are his two major books. If you can flash them on screen, that'd be good. If not, it's all right. But Dr. Stadler received his PhD in medical engineering from the Harvard MIT Division of Health Science and Technology. He's been a scientist in the medical device industry for over 20 years, and literally millions of his cardiac devices are implanted in millions of people worldwide. He has also been elected Fellow of the American Institute of Medical and Biomedical Engineers. He has obtained more than 140 US patents and has over 20 peer-reviewed publications. But anyway, that's enough from Standing and I. Rob, thanks again for being here. If there's anything else that you would like to add for your introduction, or if I said anything wrong, let us know.
Well, thank you, gentlemen. It's great to be here today. I appreciate this opportunity in hopes of reaching people out there. Thank you.
Right.
No problem. We are excited for this, guys. Check out the screen. And we've also got the links in the description box for The Scientific Approach to Evolution and The Stairway to Life. Both must-read books, guys. So why don't we get right into it? Actually, I should say before I forget, anybody in the audience, if you do have any questions pertaining to the topic at hand, we're going to be discussing Rob's book, The Scientific Approach to Evolution. Tag me. I'll field the questions that are relevant to the topic, and I'll, whenever there's a moment during the interview, I'll be sure to ask Dr. Stadler. So let's get right into it. I'm excited, Rob. The title of your book suggests that evolution has not been approached scientifically, even though many scientists support it, and that schools are not teaching evolution scientifically. What do you hope to accomplish with this book, Rob?
Yeah, I think that question is very pertinent to what our society is going through right now. And this comes up with the election and with COVID. I think society is going through what I would call a crisis of confidence in science. It feels like a lot of society is kind of turning their back on science or giving up on it. And that comes from the way we're handling COVID, the way we deal with climate change, the way we explain evolution, we explain our origins, and how it's taught in schools. And I'm not going to get political. I think there are many reasons why this is happening. And I'm talking about kind of an increasing divide between what science is saying and what the general public seems to agree with or adopt or believe in. And this growing rift, I think, is very concerning to me. And there are many reasons for it, I'm sure. But one of the reasons that I think is a big contributor to this, and it goes back to this book, is that we don't have a great appreciation of how to apply a level of confidence to some scientific evidence that we hear. You hear it through any kind of media, you hear about some evidence, and you should be able to frame that as to how much confidence does that piece of evidence provide? Is it a low confidence kind of result, or does this really tell me something that I can trust in and it's a high confidence result? And I think often we're getting that upside down, and we'll talk more about that as we go on here. But the book goes through how to sort of judge the level of confidence that some scientific evidence provides. And then the book applies that to evolution. And so one of my goals in writing this book is to improve the scientific rigor in the study and the teaching of evolution. And I really want to try to reduce the bias that leads us, it risks leading us away from the truth so that we can actually get to the truth. So we want to open up science to be able to follow the evidence wherever it leads, and not put the blinders on and keep us from being able to see the truth. That's the main goals for the book.
Oh, that's true. That's so true. Those blinders, man, if people could look non-biasedly at the evidence, I agree. I have a question though. I'm curious, what led you to write the book in the first place?
Yeah, it's not a book that I had intended to write. It kind of, it's my experiences kind of prompted me to do it. So it started with my career in medical research. So as you said, I've been doing medical research for more than 20 years. And in doing that, this is not something I was taught in school. You get it through the experience. But in our society, medicine is life and death. We take medicine very seriously because your life literally depends upon it. And in my career in medical research, you have to have a very high confidence level of evidence in order to get a medical product or a drug or something like that approved for human use. And that's because society demands very high confidence before I take this life and death treatment. So I got used to, in my career, I got used to what is required to produce this sort of high confidence evidence. And this is required by FDA in the United States or whatever regulatory body in your country. But it surprises me as a scientist then when I look outside of medicine, it's not very often talked about, different levels of confidence or different degrees of evidence. If you Google the term levels of evidence, something like that, you'll end up with all these articles about medicine and about how different drugs have a certain level of evidence. Maybe it's a 1A or a 2B or something, and they're graded as to how much supporting evidence there is for the benefit of a certain drug or a certain treatment. And so in medicine, it's very well established, but not so much in other fields. So I'm used to that. And then my son went to high school biology class, right? And I knew there would be, some challenges to that. But I read his textbook in public school and high school biology. And because my mind is already focused on these different degrees of evidence and the high levels of confidence that I have to get a medical product approved, when I read my son's biology textbook from that frame of mind, it really hit me. It was quite a surprise because everything in that biology textbook, which is trying to convince the students of the validity of evolution, all of the evidence presented in there actually met none of the criteria that I myself am held to as far as quality of evidence or level of confidence in evidence. And it was exactly the opposite, that they presented very low quality or level of confidence kind of evidence. And that really kind of flipped a switch in my brain. I said, I got to write a book about this.
That's awesome.
Well, you mentioned in the book that medicine has long appreciated different levels of confidence in evidence. This concept of levels of confidence or levels of evidence is not commonly mentioned in other fields. Why is that?
Yeah, I pondered that, and I think what it comes down to is that medicine is life and death. And if you go back a few hundred years in history, medicine was a mess. people were putting leeches on to draw blood out and try to balance your humors, and they were giving, having mercury as a treatment to give you mercury, and all kinds of crazy bad things were going on, lobotomies, because it wasn't regulated. There wasn't this scientific method, and society kind of rose up and said, we've got to stop having medicine hurt us. We want medicine to help us, right? And that placed these criteria and this appreciation for good science and high quality, high confidence kind of evidence. And I feel like other fields are coming along that route, but because other fields are not really life and death and not as litigious as medicine, right? So in the practice of medicine, if you do something wrong, you're going to be in big trouble. You'll lose your license, you'll get sued. So there's a lot of pressure to get it right there, and that's driven it. And I think other fields are going to come along eventually in this way.
I can see that. Yeah, I know exactly what you mean, because as an herbalist, I don't have that rigorous thing that you're talking about. For example, if I take something like a senolytic activator, most people have never even heard what it is, but it's something that doesn't have to go through any peer review. They can just say, oh, an onion is really good for killing parasites. And you're like, oh, okay, well, how? You can't find out how because there's no actual rigorous thing going on. So do the 6 criteria you mentioned apply only to medicine?
Yeah, so we're about to go through that. Real question is, how can I see some evidence that someone's presented to me and give a quick judgment of it? Is it a high confidence, or is it medium or low? And it's a spectrum. It's not just high or low, but how do you judge where it is on that spectrum? And we'll go through these 6 criteria that are in the book of how you make that decision. Should we go through that now then? Those 6 criteria?
Yeah, I think that would be a good idea. As to Rob, how exactly can we determine the level of confidence that science provides? And I'm going to put a visual up for the audience, actually, that'll help them understand. I think that would be a good way to approach this, Rob.
Yeah, thanks. That's great. And so in my career and in thinking this through, I kind of boiled it down to these 6 criteria, which I hope are relatively simple, relatively easy to apply. And I think, this is good for everyone to practice. Like the next time you hear a news story, some kind of scientific finding, something about COVID, whatever it is, to apply these criteria. And it gives you a feel for the level of confidence that result provides. And again, it's not black and white. Like here we have on the left, the 6 criteria to give you very high confidence. And on the right are 6 criteria, the opposite, that only give you very low confidence. It's not intended to be black and white. It's a spectrum. But working through these 6 things will help you figure that out. So I'd like to explain these by way of examples, right? So in science, you begin by asking a question. You ask a question that hasn't been answered, and then you use the tool of science to try to answer that question. So you can even tell just by the question you ask whether or not it can be answered with high confidence if the science is done right or not. So to give an example of something that can be answered with high confidence, so this is the left side of the screen. Let's ask the question. Let's say I come up with a new drug, and I claim that this drug can cure cancer. Right? That's pretty relevant. It's a life and death question. It turns out if we ask a question like, does this drug cure cancer? That question can be answered if it's done right, if the science is done right, with very high confidence. And we'll go through that. And then to contrast that with a question that cannot be answered with high confidence. So an example of stuff on the right side of the screen, let's ask a question like, can the fossils found in the fossil record, are those life forms explained by a process of evolution? In other words, did evolution produce the life forms that we see in the fossil record? Right? That's a question, and that question cannot be answered with high confidence. It's quite the opposite. So let's go through these in turn. So number one is repeatable, right? If I take my cancer drug and I need to try to prove that it cures cancer, so I could give that drug to thousands of people in a clinical study, and I can get a result, right, to show that it did help or didn't help. And maybe you don't trust me, so you can do the study. You can repeat my results. And maybe somebody in another country tries it, right? So the more the result is repeated, if you get the same answer, you build confidence, you have higher confidence, you trust what comes out of it, right? That's so obvious. Everybody agrees with that. So on the opposite side, though, here we're saying, are the life forms that we find in the fossil record produced by a process of evolution? Well, it's not repeatable. You simply cannot repeat the process that you think produced those life forms. You can't repeat the process to make dinosaurs and make all the other types of things we find in the fossil record. It's simply not repeatable. Now, some people counter that when I make that statement. They say, hey, I can find a T-Rex fossil, and then I can find another one, and I can find another one. So that's repeatable. And I say to that, yes, you can repeatably find fossils, but that's not answering the question I'm trying to answer. I mean, what you've done there is repeatably answer a question of, can I find a T-Rex fossil, or did T-Rex exist at one time? Yes, you can repeatably answer that. But that does not address the question of how those life forms, how that T-Rex came to become a living thing, right? You can't repeat that. So that's number one on the list. Number two is to have direct measurements and accurate results. So with my cancer drug, right, I do this clinical study, I give 1,000 people the drug, I can directly measure who survives, right, and who dies from cancer. It's really quite simple. It's kind of obvious who survives and who doesn't. So that's very direct. On the contrast side, back to our fossil question, what we're trying to do with that is to determine if evolution is the cause of what started these life forms, right? What created these life forms. But what we have is fossils. We've got pieces of bones. We have evidence of death, the remnants of living things. And you're trying to use those remnants of living things to try to extrapolate or try to understand what caused the living thing to become a living thing. And that's extremely indirect, right? To try to study a process of creating life by looking at death and sparse remnants of death is very indirect. And there's a lot of extrapolation. So that's quite the opposite. It's the polar opposite. Number three is a prospective interventional study. Now, those are kind of fancy words. What that means is that good science is done by designing an experiment in advance, plan it out, write it down on paper what you're going to do, and then you conduct the experiment. That gives you higher confidence if you do it that way. And why that does is because it allows you, by planning the experiment in advance, to block out anything that would get in the way, anything that would confound the results. So for my cancer drug, I can plan to have 1,000 patients receive this drug and 1,000 other patients that receive a placebo, which is a sugar pill, right? It has no effect. And I can plan the study so that the doctors don't even know which pill they've handed out. And the patient doesn't know what pill they received. It's called double blinding. And that's a way of blocking out biases and blocking out any kind of anything that could confound the answer. And I can make who gets the sugar pill versus who gets the cancer pill. I can make that a random assignment, like use a random number generator to assign that. And that helps to block out anything confounding, like, oh, what if there's more smokers in the group that gets the sugar pill, and therefore more people died in the group that got the sugar pill? That would confound and confuse the results. It could lead us to adopt a fallacy, to accept something that isn't true. So you can block all that out if you design it prospectively. And on the opposite of that, back to our evolution example, we have an extremely retrospective process. We're looking back in time, far back in time. We certainly can't prospectively turn a dinosaur into a bird or something like that. So we look retrospectively, and you cannot control anything that's going on there because it's already done, it already happened. And so you're really not sure what is influencing what you've got here. So a nice mantra I pull out of this third criteria is that if the study is done right, a prospective study can actually establish cause and effect, right? So I give the cancer drug, the effect is that cancer is wiped out. And if I do it right, I can establish that as a cause and effect kind of relationship. But if all you have is retrospective study where you're just observing things that happened in the past, you can't possibly establish cause and effect. All you can do is sort of hint at an association. You know, there seems to be an association between this and this, but I'm not quite sure what's going on because I couldn't control it. I may not even know the conditions of what happened so far back in the past. So we're halfway through. That's number 3. Number 4 is to be careful to avoid bias. And we all have bias. I have bias. Everyone has bias. And it's good to admit we all have bias because it's a fact of being human. And that bias can confound you. It can keep you from getting at the truth. So in the example of the cancer drug, where I'm trying to determine if this pill can cure cancer, I got to avoid bias. In fact, every clinical study that I design, I have to have a paragraph or two in the protocol that talks about how I'm going to minimize bias in my study. So I already mentioned with the cancer drug, I can randomize patients to who gets the drug versus who gets the sugar pill. And I can blind the doctor so they don't know who's getting the drug or the sugar pill. I can blind the patient so they don't know. And this takes away biases, even biases that are subliminal that you don't even know are happening. And so this is why, you know, a cancer study can be done with high confidence. You get that stuff out of there. On the opposite side, we have this evolution experiment, or we're trying to explain how life forms came to be. There's inherently a lot of bias there, and everybody comes to that with their preconceived belief. And in that particular question, I think the influence of bias can actually be greater, more influential than the actual data that you can find. The evidence that you dig up can have sort of less of an influence than the bias. The bias predominates and can blind us from the truth. And you can't block it out. You know, you just can't do that with that kind of a study. Number 5, we're almost there. Number 5 is to be careful to avoid assumptions. So assumptions are ways, they're kind of like shortcuts, you know, or ways of filling in for something that you don't know in a study. And sometimes you're trying to save money, you're trying to make the study go faster. So you say, hey, you know, why don't we just assume that smoking cigarettes doesn't matter in my cancer study? You know, and that would be a bad assumption. But assumptions are always made to try to make it faster or cheaper. So you really want to avoid those assumptions. And a well-done clinical study on a cancer drug, you know, if they have any kind of assumptions like that, they're going to list them out and be candid about them. They're not going to hide them. But they design the study so that you avoid assumptions. On the other side, with our question of evolution and the fossil record, there's many assumptions baked into this. And a couple of them that are my favorites are that, you know, if you're digging through layers of sediment, and in a given layer from a certain age, if you don't find a particular kind of life form, then that means that life form didn't exist at the time that that layer of sediment was produced, right? That's a typical assumption that's made. You can't really prove it, but you assume that's a fact. But then some evolutionists will actually make the counter assumption, and they'll say, well, we know that there are very few transitional fossils, right? And if I can't find anything transitioning to become a bat, for example, and that's kind of a fact right now, nobody has found anything that seems to have been a transitional form to get to being a bat or a fish or whatever. They assume that just because you can't find that transitional form in the fossil record, that doesn't mean it never existed in the fossil record. So So those two assumptions are actually contradicting each other. But there are many assumptions going on when you try to interpret what a bone is doing buried in the ground like this. And that's dangerous. That leads to low confidence. And then finally, the last one. The last one is a little bit of a different criteria, but this is really important because in science, eventually you have to communicate what you come up with. You got to share your results and publish it or communicate it somehow. And how you frame it, how you describe what you came up with is so important. And you can tell just by the way people summarize their results, if they're overstating the confidence that they found, if they're stretching the truth even. So when I finished my cancer drug study and I published the results, maybe I would say something, if I had a conclusion like, this drug cures cancer in all humans, every kind of cancer, and I'm kind of slamming my fist on the table saying how awesome it is. You could tell that's probably not exactly the case. And then you read my study and you find, oh, I only studied thyroid cancer, and I only studied females, and I only studied people in the United States. So if I'm going to present my results with sober judgment, the concluding line should be something like, in females in the United States who have thyroid cancer, this study suggests that this drug is effective in curing cancer, right? That would be a sober way of concluding what I've actually done and what you can take home from it. The other result where I say, hey, this cures all cancer in all people all the time is an extrapolation. It's overstating confidence, and it's kind of going beyond the scope of my study, and that's really bad science. And you see that a lot when media picks up on results, and they kind of amplify and hyperbole and stretch the truth a little bit. So number 6 criteria when it comes to this fossil record, our biology textbooks, they'll say with great confidence in the statement that all living things have common ancestry, and the fossil record proves that, life forms descended from each other through common ancestry through the process of evolution. And they don't put any kind of hedging in there, like we think so, or evidence suggests that, or, they state it with very high confidence, which is an artificial kind of confidence, and it's bad science. And that deeply concerns me. So the next time, just summarize this, the next time you hear some evidence on some media, some paper you read, whatever it is, you can apply these 6 criteria to what you see and get a feel for the level of confidence that result provides. And that's really one of the take-home messages from this book, is to be able to do that.
Well, I think those were some great points, and I can't recommend your book enough. And it's true, they like to treat, for example, the fossil record as high confidence science, right? And for all the points that you brought up, Dr. Stadler, this is precisely why I prefer high confidence empirical science. I like the way you put it. We like to avoid bad assumptions. We like that which is measurable, as you said. When it comes to drugs, we can find a solid result through repeated studies and, say, giving the drug to thousands of people, where we can build confidence in the science, as compared to, and I like the way you put it, as compared to fossils. They're not repeatable. We can't repeat this process that produced these life forms in the fossil record, right? I think this is why, Rob, I find evolutionists, when I interact with them, they often like to hang out in the fossil record in terms of arguments, because it's so low confidence. There's so many stories and interpretations that can be put forth based on such indirect evidence, right, as compared to that which is direct, as you stated. So great points.
That's right. And you can make whatever story you want. And because the evidence is such low confidence, you can argue forever and ever and ever and never get to a resolution. And also, because it's low confidence like that, it can be very difficult to falsify. It can be difficult to prove that you're wrong.
Right.
If I pull out a bone and I say this bone is transitional between dinosaurs and birds, it's pretty hard to prove that wrong. And I can make up a story and if you can go back to the screen for,
Yeah,
I want to give a nice sort of object lesson to it.
of
For me,
course. Yeah.
and it's important to know that some fields of science cannot really be practiced with this high confidence, these criteria for high confidence, right? If I want to study, let's say I want to study if living next to a nuclear power plant decreases your intelligence or your health or something like that, right? To do that right, to do that with high confidence, you would design the study in advance and you would assign some people to live next to a nuclear power plant, some other people you assign to live far away, and you study them throughout their entire lives, and you control everything else so that they live the same life, except, the same food and the same hobbies or whatever it is, except some people live near the nuclear power plant and some don't. That way you could get a very confident answer, right? But of course, that's just not ethical. It's not practical. It's not going to happen. So you can't study that question with real high confidence. And that's okay. That's the best you can do. But just don't try to tell me then when you get your results, don't try to tell me these are high confidence results, because to be honest, to be humble about it, you've only gotten low confidence results in what you've done. Now, if you're stuck there with low confidence science, the analogy I like is a drinking straw, right? This is a simple drinking straw.
Yes. Yeah.
So imagine that you have to referee a football game. It's now football season, so you can picture yourself being the referee. You're under a lot of pressure. You got to catch the play. You got to catch people who are cheating, right? Whatever's going on down there. But you're the referee. And now in a low confidence world, instead of being able to watch the game with your eyes, you have to close one eye and you can only look through the drinking straw with the other eye at the football game, right? And so you can see a little bit of what's going on, you get kind of a foggy picture of a little bit of what's going on there. And that's what low confidence science provides. And you have such a hard time to see the big picture and arrive at the truth. That's what it's like.
Yeah, I really love that analogy. The second I read it in your book, I thought, this makes such sense. It makes a lot of good sense that's especially with the fossil record. They're not looking at the bigger picture. It's like they've got a straw and they can't see anything in their peripherals. I like the way you put it. I guess if they were to, because the way you said it was, it's oftentimes the best they can do. It's the best they got. So if they were to just humble themselves and admit, okay, this is the best we can do with, say, the fossil record, for example. Yeah, it's low confidence, low quality science, but I think it just comes down to humility as compared to the way you said it. They treat it like in the textbooks in school, they treat it like it is high confidence science. It's irrefutable,
And I also want to clarify that, low confidence science doesn't mean you're a bad scientist, you're a bad person or anything like that.
Yeah.
In a lot of cases, if you're an anthropologist or whatever, you're trying to do the best you can. And you are doing the best you can. The problem is all you've got is a drinking straw and you're trying to look through it. And that's okay. It doesn't mean you're a bad person, but just as you said, with humility, just say, hey, this is the best I could come up with. And, we can't be certain, but here's what we got. That's fine.
Yeah, and you make that point in your book, so I really like that. Okay, we're getting a lot of good feedback from the audience on that. And this next question has actually been a question in the audience. So, Rob, do the 6 criteria apply only to medicine?
Yeah, thanks. I get that question a lot. And I try so hard to explain that they apply to all fields of science. These 6 criteria you can apply to any field of science of any kind, and I think they're equally applicable. It just so happens that medicine, is able to do high confidence science, and other fields of science may not, like anthropology, may struggle, but it still applies. It applies to all science. In my book, I go through the example of just simply studying how fast an object falls when you drop it, and that can be studied with high confidence science. It has nothing to do with medicine, and it applies. So it's on all fields of science.
Yeah, well, I like what you said earlier about how it makes it nearly impossible to falsify, right? Because remember, the slow Darwinian process of life evolving slowly. Well, when they find something that didn't evolve, boom, it's stasis. It's no big deal. Or when they find something out of place that shouldn't be there, oh, it's punctuated equilibrium. It jumps forward massive amounts of time. So it makes it unfalsifiable. That's not very good.
Exactly.
You mentioned in your book, specifically on page 52, you talk about a TED Talk with paleontologist Jack Horner about finding and naming new species of dinosaurs that show a common error when we rely on this low confidence evidence. Can you explain that for everybody, please?
Yeah, I would recommend people watch that video. You can look it up on YouTube, Jack Horner TED Talk. But what's fascinating there is that you have paleontologists, right? And a paleontologist is trying to build their career like anybody, as a scientist, trying to make a name for themselves, trying to make a discovery. And so they discovered dinosaurs. They dig up something and they say, well, I dug up something and it doesn't look like the other dinosaurs that have already been named. So it's something new. It's a new species, and I'm going to name this thing and put their stamp on it, write a paper, and become kind of famous. So Jack Horner looked at this, and he realized that all the dinosaurs that are out there, the fossils, were claimed to be, they were adults, basically. And he actually looked at the dinosaur bones and was able to get permission to drill into the skull. And apparently when you drill into the skull and you look at the bone, you can determine the age, the maturity level of the animal when it died. And so he was able to piece this together that an infant, a toddler, an adolescent, and an adult dinosaur of the same species were named for different species because the adolescent didn't look like the adult and the infant didn't look like the adolescent. So people were naming them as different species when in fact it was the same species, just different ages. And he said this happens because people like to name things and they like to be successful. And basically what it comes down to is that bias, that you really want to discover something new. You want to be famous. And so here it is, I found it, I discovered it. And that bias can lead you to adopt a fallacy, right? To believe something's true that isn't true, all in the name of science. And so knowing that that's happened, and it's cool that Jack Horner was able to clarify and get to the truth in this case. But knowing that can happen, knowing that dinosaurs can be thought to be separate species when they really aren't, then if someone makes a statement like, through the fossil record, this transition to this, transition to this, transition to this, right? That's the story that we're told. How do we know that that's not just bias, right? In the same kind of sense. How do we know it's not the same thing going on there? And it's all low confidence, and it's kind of cloudy. You're looking through the drinking straw. It's pretty hard to get to the truth.
Well, I find that amazing, Rob. I've mentioned that specific case a number of times now after reading that in your book, that specific case. And as you put it, there's a lot of pressure in this field to find that next big find, right? Because not only that, I've read a ton of papers and watched some lectures on the hominid fossil record itself, and there being numerous cases of mixed bone beds, and then as a result, chimeric or artificial species where you've got a mix of like Australopithecus bones and human bones, and the paleoanthropologists are admitting this. So it's just the nature of what you're saying, right? It's just the nature of it being low confidence.
And it's okay that that's what it is, as long as you openly discuss that, hey, these are the assumptions we've made. Here's some limitations to what we've done.
Yeah, I think those are great points. Exactly. I completely agree. So, Rob, you mentioned earlier your son's high school biology textbook. What evidence was presented for evolution, and how did it stack up against the criteria for high confidence?
Yeah, so, we have to start by recognizing that in the public school biology textbook, sadly, it's not so much about science as it is about salesmanship. It's like when you go to the car dealer and you pick up a pamphlet about some car, right? It's going to talk about the great qualities of this car, how awesome it is, and all that. It's not going to say something like, well, it's true that it doesn't get the best gas mileage, and yes, we've had transmission problems in the past, but it is a pretty good car, right? It's a sales argument. And so the biology textbook expectedly is, unfortunately, a one-sided argument that's trying to convince kids about evolution. But when I look at the evidence that was presented in there, and I've seen many biology textbooks very similar, but they largely rely on four pieces of evidence. And first is the fossil record. So we've talked about that. It's very much low confidence. Next one is homology, which is to show that across different types of living organisms, we have similar genes or similar bone structures, like we have 2 long bones here and 5 phalanges and so forth. And you'll see that in humans and in bats and in fish or lizards. So that's homology. The third would be vestigial organs or bad design in living creatures. That's an argument. And the fourth one is biogeography, which is how different living organisms are dispersed around the planet, or some only live in certain regions. So in the book, I go into all the details. We're not going to do it here, but each of those 4 types of evidence, each of them fails all 6 of the criteria. So it's not just lowish confidence. It is kind of like the lowest confidence of evidence in each of those that the biology textbook praises as being convincing. Unfortunately, that's the situation we're in, and that's what they emphasize.
Right.
Great answer, Rob. A couple of relevant questions that, before I miss them, because once there's too much going on in the chat, I'll miss the question. So this one came in the form of a super chat. Doki Doki Bible Club, thank you so much. Asks, is RNA world high or low confidence? I think we know the answer to that one.
Well, good question. So first, RNA world this is relating to origin of life, right? To get the first living thing started. And of course, cells are extremely complicated. And so there's a hypothesis that life began first with just RNA, because RNA is able to be a catalyst like an enzyme, and it's also an information carrying molecule. And that's why there's some excitement about RNA world. But if you look at those 6 criteria, right, we can go through it real quick. RNA world is not repeatable, meaning we can't go back before there was life and the conditions that existed and try to repeat an RNA type world. You can try to do it in a lab today. You can do it in your little flask with your chemicals and your hoods and all that stuff. And you can try, but you're not exactly repeating what happened back then. You can't be sure of what happened back then. Number 2, it is very indirectly measured. I mean, we can't even go back in time there. We don't even have any real fossil evidence from supposedly 4 billion years ago. Nothing is left from back then. So it's extremely indirect. It's extremely retrospective to go back, unless you do it in the lab. And if you do it in the lab, you've got tons of assumptions, which is number 5, many assumptions required. There's clear opportunities for bias, because the people who tend to promote RNA world are trying to promote a naturalistic explanation for life. So quick answer, it is very low confidence, yes.
Great answer. And one last question here that is relevant to the point you just made on the textbook examples. Question here, org 343, should those 4 textbook examples be more thorough or removed?
Well, great question. I think they should be explained with a sort of even-handed, unbiased statement that, hey, there may be some hints here. Because this is low confidence evidence, there's some hints, and it's possible to interpret those as supporting evolution. It's also possible to interpret it a different way. But what should be done, though, is to take that low confidence evidence and deprioritize it and then take the high confidence evidence and prioritize that in its place. And we haven't gotten to it yet, but there actually is some quite high confidence evidence and experiments that speak to evolution, and those should be put up high in their place. If I go to FDA with my cancer drug and I say, hey, I've got a randomized controlled study of 2,000 people, blah, 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 I did everything right and it's high confidence, and here's what that result found. And, oh, by the way, I actually also did a retrospective observational study where some people bought this stuff on their own choice and ate it as a dietary supplement, and it looked like it kind of helped. You know, if I do that, the FDA would just blow off the low confidence results, wouldn't even look at it, wouldn't even think about it, and they focus on the high confidence evidence. And that's what we should be doing when we talk about evolution.
Absolutely. Well, given that your book is rather discouraging, to say the least, to those who believe in evolution and molecules to man, it may be surprising to some of our viewers that your book has a chapter called High Confidence Evidence for Evolution. So what is exactly in that chapter?
Great. Yeah, this is where there's a twist in the plot. So there actually is a fair amount of data, and it's building every day, of evidence that meets all 6 of those criteria that teaches us about evolution. So let me give one simple example, right? Is often cited is evolution of dogs, right? So we know over the last few hundred years that wolves, through a process of artificial selection, have given us this great diversity of dogs that we have today. And it's pretty cool, you know, incredible diversity of the types of dogs. They're all kind of the same species because they can interbreed, but it's pretty awesome. And that's what, you know, Richard Dawkins makes a big deal of that. And if you look at that study of dogs coming from wolves and you look at the 6 criteria, you can argue that it meets all 6 of those criteria. You know, you could do it today, take a wolf and start breeding and artificially selecting, and you could end up with a Great Dane or a Golden Retriever, you know, over hundreds of years. You could do it, you could repeat it, and you can meet all 6 of those criteria. So that's high confidence evidence, but it's high confidence evidence of, you know, what you call microevolution. So in the book, I love this quote from Richard Dawkins. You all know Richard Dawkins. And this is from The Greatest Show on Earth, which is his book about evidence for evolution. And he talks about the dogs, right? And he says, this is his quote. He says, it's on page 82. He says, in 82 in my book, he says, if so much evolutionary change can be achieved in just a few centuries or even decades, just think of what might be achieved in 10 or 100 million years. So what he's doing there is asking you to extrapolate. He's saying we have a result here, which we don't argue about, you know, that lots of changes have happened in 200 years. He's asking you to extrapolate that for 10 or 100 million years. That is something that nowhere else in science is that allowed, where you can observe something here and extrapolate that 10,000, 100,000, a million fold down the road and have any belief or any confidence in that result. It's unthinkable in science that you could extrapolate like that. Now, if you look at those 6 criteria of high confidence, the dog coming from a wolf, you can meet all 6 of the criteria for high confidence. In that quotation from Richard Dawkins, where he's asking you to imagine going forth 10 million or 100 million years into the future, that statement immediately contradicts all 6 of the criteria. He's asking you to go from high confidence to the absolute lowest of confidence and to trust him that that's what's going on here. And the ultimate irony for me is that in creating that, in that statement he made, which is just unthinkable bad science, what's ironic to me is that before Richard Dawkins retired, his job title was the University of Oxford's Professor for the Public Understanding of Science. So he was recognized as being the guy who's going to help the public to understand science. And yet he did something like that, which is extremely unscientific, very disturbing. So that's just one example of high confidence science that speaks to evolution. Let's go through a couple more. We have Richard Lenski's experiment. Some of you, probably most of you, are familiar with that, right? He's up to like 72,000 generations of E. coli in a flask. This is done repeatably because he's got 12 flasks, and you can also freeze and thaw, parts of the experiment. You can repeat what happened. It's very repeatable. It's directly measurable. He can look at the DNA at the beginning, look at the DNA 70,000 generations later, and directly determine what's different about the DNA. It was prospectively designed. I don't think there's much bias or assumptions, so forth. This is very high confidence science. It's very well done. And it tells us, what is evolution capable of. And one more case that I really, really like this case. It was done by Ann Gager, published about 10 years ago. This is an example of high confidence science applied to evolution. And in this case, they took E. coli, and E. coli is able to manufacture an amino acid called tryptophan. It has an enzyme that produces the tryptophan, which is great. So Ann Gager said, hey, let's do an experiment. I'm going to intentionally damage that gene. I'm going to intentionally damage the gene so it produces an enzyme that's half, broken. It's not very good at producing the tryptophan. Put that back in the bacteria. Then I'm going to put the bacteria in a bath, in a flask that has only a limited amount of tryptophan. And so it better be able to fix itself in order to survive and grow, right? Now, the way they damaged the gene was just changing one nucleotide in the DNA. They changed one nucleotide, which changed one amino acid in the enzyme. So a very minor damage. And they found that after having about 1 million bacteria produced, no, it was like 100 million bacteria produced, that actually a random mutation actually flipped the one nucleotide back to the way it should be. And the enzyme was fixed. And the bacteria could produce tryptophan, and it was able to grow rapidly and survive and dominate. So there you have in action, right? High confidence, because it can be repeated, it can be directly measured, blah, blah, all 6 criteria. And you have evolution to change one nucleotide to make a better bacteria. Okay, that's cool. Then they did the next step, and they said, okay, let's challenge it with 2 nucleotides changed, right? They had the first mistake, they added that, they added a second change to that. So now there are 2 nucleotides changed. That produces a tryptophan synthase, the enzyme, that is completely broken. It doesn't work. But all the bacteria has to do is change 2 nucleotides to make it fully functional. And in fact, if it changed the first one, if it fixed the first one, it's somewhat functional. It's a step in the right direction. And then it needs to change the second one to be fully functional. So this was a test. Hey, evolution, I'm giving you a test. Can you solve the test? So they put that in the bacteria, put the bacteria in a solution that contained a small amount of tryptophan, and they went through 9,300 generations of the bacteria. 9,300 generations, and something like 1 trillion bacteria organisms formed through reproduction. So there's plenty of opportunity there in 1 trillion organisms to have a fortuitous mistake to make it better again. And they found not only did the E. coli not fix the problem, so it was not able to repair the tryptophan synthase enzyme. Not only was that the case, but in the 15 flasks that they tried, the repeatability of this, some of the flasks just jettisoned the gene entirely, or they inhibited production of the enzyme because they're better off. The bacteria were better off not even trying to produce that enzyme because it's a waste of energy. It actually takes tryptophan to make that enzyme. So they're burning their tryptophan they do have. So it was easier to just give up rather than try to fix those two nucleotide errors. So this gives you a feeling of the bounds of how far evolution can go. You know, Mike Behe describes it as the edge of evolution. How far can evolution go? And this was a prospective test to see how far can evolution go, and it failed the test. And this is high confidence. It meets all 6 of those criteria. So there is high confidence evidence that addresses evolution, and it's way above the kind of evidence that you'll find in the biology textbooks.
Yeah, that's awesome. So many great points there, Rob. It would take us all day just to kind of touch on all of them. That was an answer everybody needs to hear. Very important. Just like Richard Dawkins. Most evolutionists, they commit an extrapolation fallacy. I like the way you said that what they're doing is they're extrapolating the data. Just imagine, all these small micro evolutionary variations over millions of years. Imagine what could result, fish to fisherman, right? But what I find funny, Rob, is the fact that the changes we see are typically downhill and reductions in allelic variability. Like you were talking about with Lenski's experiment. What Lenski has actually observed at this point, from my understanding, is that his bacteria overall have shrank in functional genome sizes. There's been no real uphill changes. So what comes to mind is, okay, you want to take that extrapolation fallacy back millions and millions of years. Well, if all we see is reductions in information, loss of information, well, that's not really going to take your fish to fisherman. So their high confidence science doesn't help them at all, would be my thoughts. So a lot of great points there, Rob.
And I believe that's why they end up kind of de-emphasizing this high confidence evidence, and they emphasize the low confidence evidence and put that first and foremost. It's all backward.
Great point. Really good point. Okay, in the next chapter, Rob, you apply the findings from high confidence evolution experiments to the hypothesized evolution of humans from primates.
Yeah, so here we kind of get to the punchline, right? So if you do it the right way, which is to de-emphasize the low confidence stuff. So let's get rid of talk about the fossil record and homology and vestigial organs and just push that aside. Let's talk about taking these high confidence studies like Lenski, like the one I mentioned about tryptophan with Ann Gager, like dogs. There's other studies on fruit flies. And we know how much evolution can accomplish in 70,000 generations. We know how much it can accomplish and what it can't do. So let's put this to practice now. And let's talk about humans. Did humans evolve from primates? Right? And it's time to apply that. This is kind of a reality check. So humans and chimps, we're told, have a common ancestor that's about 6 million years ago. The problem is that humans and chimps are very slow to reproduce. We have 2, 4, 5 offspring, and we do that in 20 years or 15 years, maybe 12 if you're really fast. So in 6 million years, you could tell how many generations are out there. And you can also guesstimate how many humans have ever lived ever, right? You can guesstimate that. And it's nowhere near how many bacteria that I can study in an experiment in my lab. I can easily surpass the number of humans that ever lived, just doing Lenski's experiment. He's got trillions and trillions of bacteria in his study, and he's got 72,000 generations, which if you do the math, that's almost 2 million years of human generations, right? So you just take what Lenski has learned and apply that to humans coming out of chimps. And I think that's a very interesting result because, you've had this many times on your show already, but we're all told that humans and chimps are 98% the same DNA. And I know you guys have gone through this many, many times, but with the latest genome sequencing of chimps. I think it's called Pantro-6. The true number of the agreement between human DNA and chimp DNA, I think, is now in the mid 80s, something like 80%, certainly not 98%. But it almost doesn't even matter, because even if it is 98% similarity, and we have about 3.2 billion nucleotides of DNA, you're still talking about tens of millions of nucleotide differences between us and chimps. And we also know what's called orphan genes. I think you guys have been through this many times on your show, but every species has orphan genes, which are unique genes to them that no other living thing has, that make them special and unique, right? I think human orphan genes are going to give us big brains, consciousness, ability to speak and communicate. So there's a lot of interest in these orphan genes that we have and chimps don't have, nobody else has. If you just look at those orphan genes, you read the papers about this, there's something like 1,400 orphan genes that we have that chimps don't have. So when you add this up, you're looking at, you need to come up with 1,400 new genes, you need to come up with tens of millions of nucleotides of DNA between us and our common ancestor with chimps in 6 million years. And then we look to Lenski's experiment, this one by Ann Gager, and all this other high confidence science of evolution. And you end up with a huge, what's called Haldane's dilemma, or the waiting time problem, that there's just not enough time. There's not enough generations. There's not enough human beings ever that existed to accomplish the kind of changes that would be required.
Well, so many good points there, Rob, and some of our favorite topics. You're right. We could probably spend a couple hours on each point. And I constantly hear on that last one, the waiting time problem, you'll hear the critics, constantly repeat that, oh, it's been solved. And I typically respond with not even close, because from my understanding, in addition to the single letter differences, you're looking at, I've read about tens of thousands of chromosomal rearrangements would have had to occur, spread, and also become fixated as well. And tens of millions of base pairs of insertions and deletions. And what we know about the rates of fixation, how slow they are, and most new variants are lost. This is a huge, huge waiting time problem that I think a lot of the critics, or those that just kind of accept that type of evolution, they don't really think about these problems. So I'm really glad you brought those up.
Yeah, and it's easy to get single nucleotide changes. Those kind of like salt and pepper, they just pop up all over the place generation by generation. But if you need specific changes to occur in specific places in order to make a new function, or to come up with an entirely new gene that's never been produced before, we just know that the probability of that is just astronomical. And humans are so slow to reproduce, and there's so few of us in relative terms compared to a bacteria, that it just makes no sense that that's possible.
Yeah, great answer. One quick question before we move on is in regards to the topic of orphan genes that you were talking about. I find that to be a really fascinating line of evidence for independent origins, because I find that this is exactly what we'd expect to find, is these taxonomically restricted genes. How does the evolutionary community, Rob, how do they deal with that observation? What are their explanations? And I guess the best way to ask the question, are there explanations, whatever those are, for the existence of orphan genes? Would that be considered high confidence?
Good question. Yeah. So I think you guys have covered this a lot on your show, but there's two proposed explanations for orphan genes. And one is just random sequences that got lucky. And the second is that a gene gets duplicated, and then that gene changes its function over time until it's not recognizable anymore from its old twin, and has a different function in a different sequence. And the way they study those then is to do phylogenetic trees and to look at the DNA similarities and try to pull this together. And it certainly isn't high confidence because it's not repeatable and prospectively studied. You don't see these things. You can show me examples of gene duplications and then some divergence where the duplicated gene slowly changes a little bit, but you're not going to see it diverging so much that it's not recognizable anymore, which is what the theory would say, that they're no longer recognizable and now you have an orphan gene. You can't repeat that. You can't do that prospectively. So there really isn't much confidence to it, but it's the only thing they have, so they have to kind of grasp at what they've got.
Makes sense. Makes sense. One of the main points in your book actually is that low confidence evidence is being prioritized over high confidence evidence in the teaching of evolution. Why is this not recognized?
Yeah. Well, that's my main frustration, and that's the main point I want to convey is, like, can't we just do what rational scientists would do, which is prioritize the high confidence over the low confidence? It just plain makes sense. I think the low confidence is prioritized because, as we've said, you can make up a story and make that story agree with what you want to be true, to make it agree with your hypothesis. And you can promote that all day long, and it's very hard to falsify it. It's disturbing to me. And when I went back to our intro, when I said that our society is having a crisis in confidence when it comes to science, I think this speaks to it too, is that people have good intuition and good common sense. And if you try to tell them a story, and they don't see real confident evidence backing it up, they're just not going to believe you. And then you erode confidence in science, you erode confidence in whatever a scientist says, because they're calling your bluff. And the other part of this, there's another dimension to it we haven't talked about, and it's kind of the elephant in the room. And it goes by the fancy name of methodological naturalism. And maybe you guys have talked about this on your show too. But a lot of people argue that science can only be practiced under the assumption of methodological naturalism, which means that science can only address natural causes, that science is not equipped to speak of anything that is not a natural cause. And so we start off thinking, okay, how did life begin? There could be a natural reason, or there could be a supernatural, a non-natural reason. But if we're going to study this with science to try to get at the truth, which one of these is right? Well, science can't deal with something that's not natural. So that one's off the table, and we're left with the natural explanation, and that must be the truth. And so here you have what's called the truth-seeking argument, that if you apply science and you must have methodological naturalism, you're no longer seeking truth. All you're seeking is the best possible natural explanation. Doesn't matter what the truth is, you're going to come up with a natural explanation, and that's all you got because you've already blocked out the alternative. And that is, the way a lot of people want to practice science. And when it comes to origins, the question of where living things came from, there's only one naturalistic hypothesis, right? And that's evolution. That's neo-Darwinian. That's all there is. So it's the only game in town. Everything else is excluded. And so we have to find ways to support this. Science has to support this. And maybe that's what makes people do something that's very foolish, is to flip low confidence and high confidence evidence up on its head. So let's emphasize the low confidence because that can support our one hypothesis. And let's suppress this high confidence evidence that we don't like the looks of it. I think that's what's going on.
Awesome answer, Rob. We're getting a lot of good feedback in the chat. Really lively chat. Good audience today. Really enjoying this. Time's flying by. I've got a couple questions that I managed to save. in the brink of time before I lost it. Here's one here that's pretty relevant, Rob. Would low confidence work for 9th grade bio and higher confidence for 12th grade bio?
Well, maybe I'm not understanding that, but I'd prefer to have high confidence for both. I mean, no matter what age you are, what level of understanding, you got to prioritize the high confidence always. Put that on top, and you're very justified in suppressing. You can mention the low confidence with a little asterisk next to it to say, well, we're not really sure. This isn't very clear what it's trying to tell us. But that's true no matter what age you are, no matter what field of science you're studying.
Right. Great answer. Right here we got Anthony Maurice. This is from earlier. He's asking SFT, when he says they can produce RNA in the lab, it's not. Oh, maybe that wasn't his original question was, what does Rob mean by in the lab? Does this mean highly controlled environments? To summarize his question.
Yeah, right. So you can, I guess we were talking about RNA world at that time, right?
Yes.
Right. So, you can manipulate RNA in the lab, and we have to do another show on origin of life and on RNA world.
I think that might have sparked. I'll I'm sorry. Go ahead, Rob. I was cutting you off there.
Well, just to go along with it, there you do it in the lab. It's highly controlled. It's highly contrived, and it's overly simplified because you have very pure starting ingredients. A very clean flask with very pure ingredients, probably homochiral that you add in, and you can get some results. But whether that has any relevance to some dirty rock, 4 billion years ago, tons of assumptions, an extraordinary amount of assumptions would have to go into believing that that's at all related.
Right. Great response. Let me see another question here that came in, if I could find it. Like I said, lots of good feedback. Actually, as I'm looking for this next question, Matt, did you have anything that came to mind there, brother?
Yeah, I like I like to imagine that if I drank some coffee and my headache went away, and I'll be like, oh my gosh, I got to sell coffee to get rid of headaches and just patent it. You know what I mean? As fast as I can, because it worked for me one time. That's kind of what they're doing, right? I just read a study recently. I think you'll find funny. They believe that human males grow beards because of being punched in the face. That's the evolution of beard. And their theory on this was based on that if you wrap a bone in sheep wool and drop weight on it, it doesn't take as much damage as if you don't have wool wrapped around it. What kind of confidence do you think that would be in that theory?
It is. It's laughable. It's interesting. You can make any story you want to make, and it's surprising how much of that ends up being published without scrutiny. It's quite shocking.
Yeah, that was published. That's peer-reviewed, published.
Well, Matt, if that's the case, then you should have a huge beard.
Man, you think.
He gets punched a lot.
All of that cage fighting. So a question here that came in, but I can't seem to find it to screen share it, has to do with the high confidence, low confidence science. Rob, is that a new term, or is this something that's always been recognized? Was the question in the chat?
So that specific term and these specific 6 criteria, they're new in a way, but it's kind of rehashed. I mean, if you Google levels of evidence,
yes,
you'll find it's all related to medicine, but you'll find in medical terminology, you'll find the kind of criteria we're talking about in these 6 criteria. It's not the same, but it's out there, and medicine most certainly appreciates these things.
Well, it's really, your book was really fascinating and helpful to me, because I've always been on the side that, it's the direct lines of evidence that we should be using to answer these really important questions on evolution, ancestry, creation, and genetics being a really, really direct line of measurement, being high confidence science. And as you pointed out, with the changes that we see in, say, dogs or the changes we see in these experiments where you've got thousands and thousands and thousands of generations of bacteria. And all you ever get is, in those experiments, more bacteria and oftentimes more degenerated bacteria. Do they have any real solid examples in that realm, though, that could be considered high confidence for that type of evolution? Maybe like a true gain in novelty, or is that what Richard Dawkins, he kind of put forth what he thought was the best one in the Lenski's experiment, correct?
Yeah, it's kind of sad that in his book, The Greatest Show on Earth, it's on page 130 of his book, he interprets Lenski's experiment, and he made a bold conclusion about it before they actually had the DNA evidence. So there was a big change. I think you guys all know this. In Lenski's E. coli, there was a big change that occurred where the E. coli could metabolize citrate.
Yes.
Where normally they couldn't do that in the presence of oxygen, right? And before they had the DNA data, Richard Dawkins said, this is a quote from his Greatest Show on Earth book, he said, not only does this show evolution in action, not only does it show that new information entering genomes, not only does it show combinations of new genes, right? So he's sort of dreaming from his paradigm of what had happened in Lenski's bacteria. And it was about 6 or 7 years later that the actual papers came out with the DNA sequencing that showed, as you've covered on your show many times, that it was simply moving one gene to a different promoter and then changing how much another gene that existed already was amplified. So there was no new information. It was just a little gentle scrambling of some of the information that's already there, which actually made the bacteria less successful in the normal environment, but more successful in this particular environment. That's what happened. And it's very irresponsible for Dawkins to make such a strong, pound the table with your fist kind of conclusion based on his bias and his paradigm before we actually knew what actually happened. Now that we know what happened, it's pretty unexciting. So I think your question actually started with what's the most impressive, new information, new gene kind of thing that we've seen with high confidence, right? And you often hear of this nyelinase enzyme, so a bacteria that evolved to be able to eat nylon, right? I think it happened in Japan near some chemical factory. It sounds like a horror movie or something. So similar to Lenski, that was initially thought to be a whole new gene, a whole new protein with very creative, different design to it. And I think over time that's been proven to be some pretty modest, just a few changes in the DNA sequence that resulted in this ability to eat nylon.
Yeah, it's such a great answer, because when I first got into this, and I started reading their literature, just even talking to people of the other side, on the evolutionist side, they'd provide these examples. But then when you further look into them, what you see for the most part is largely reductive evolution. And like you said, even with that advantage seen in Lenski's experiment, the best you're seeing is an increase in fitness in a very narrow sense, because the rest of these genomes are decreasing in functional genome capacity. Or I just love that Lenski experiment, because I've read exactly what you quoted, Richard Dawkins saying the same thing, and yet all you see is a loss of genes. I find Lenski's own data is revealing clear evidence of genetic degeneration. So that was a really good answer.
And recently it's been published that the bacteria that develop the ability to eat citrate are actually getting pretty sick right now. They're kind of falling apart over time.
Yes. Yeah.
It's not good.
That's a really good point. I believe that was shown in a 2020 paper, which means that's as up to date as you can get for Lenski's experiment. So if what we're seeing, according to Richard Dawkins, that this is one of their best lines of evidence for that type of evolution that they believe in, that they want to extrapolate and yet that those adaptations are just accomplished by means of what? Loss of function, loss of regulation mutations. Now they're sick. I mean, that is a really good point.
And I think this is leading to this crisis of confidence in science. You know, if the public is losing confidence in what scientists are saying, it's because of things like this. You know, you got people slamming their fist and saying stuff that turns out to be wrong. And you can tell it's all coming from their bias. It's very disparaging.
It is. I couldn't agree more. And I could not recommend I couldn't recommend both your books. I recommend highly for everybody. Today, we just focus primarily on the scientific approach to evolution. So I can't recommend that one enough for the audience. It really helped me tremendously and put things into perspective. But the Stairway to Life, an Origin of Life reality check, hopefully we can have you on again in the future, because that is another one that we could easily talk about for another hour and a half. So, did you want to briefly, maybe in a few sentences, give everybody in the audience a brief summary of your book that we've talked about today, Rob?
Yeah, just a quick wrap up. If you're in the elevator and you have 30 seconds with somebody, you have to tell them something really quick, right? So I would say, simply that the evidence that's commonly used to support evolution, it actually fails to meet these simple criteria that tell us how much confidence we can have in science, right? But there is other evidence out there, these high confidence evolution studies, and those meet all of the criteria. And that evidence tells us very much that we're talking about a very constrained process that's only capable of taking a few steps in one direction, but certainly not capable of molecules to man.
Yeah, that's really good. And I want to thank the audience too. They've had a lot of good questions. And Dr. Stadler, there's one final question I have here from the audience, actually, that if you don't mind, I can ask right after this wrap up. And this would actually culminate all the questions that I've had. So this question was from Lena Powell. And she asks, has Dr. Stadler seen a computer evolution simulation? And does he think that using them as proof, quote unquote, of evolution, would that be circular reasoning?
Well, computer simulations, you really can get them to do whatever you want. They can be useful to provide some insights. I would say what they come up with can be hypothesis generating. In other words, it makes you think of an idea that, hey, we should test that in an actual experiment. I don't put a lot of trust or confidence in what they come up with. On the flip side of that question, John Sanford has produced computer simulation he calls Mendel's Accountant, and that's his simulation of it, which shows very constrained progress that evolution can make, which opposes what some other simulations have said, that they can come up with all kinds of stuff. So it's not very high confidence because it is a simulation. And of course, it also takes human intelligence to build the thing and to make it work.
Right. Great response. Yeah, no, that's Mendel's Accountant and Dr. Sanford's work on that is really amazing too. Well, I mean, we've had you for an hour and a half. This was awesome. Time has flown by. I've really gotten a lot out of this, and I really appreciate your time. Rob, the audience has loved it. Tons of good questions. We could probably have you here all day. What we'll do is we'll wrap it up here. Unless you had any final things to say, Matt, before we...
No, I just hope we can get Rob on just for a random Q&A sometime for the audience. That would be...
You can also, you could send questions to me. I have an email account that's scientificevolution.com@gmail.com.
Okay.
A little complicated, but scientific evolution, one word, scientificevolution.com@gmail.com. Send questions.
Awesome. Okay, well, I've got both Rob's books in the description box. Please check them out. Like I said, I flew through the scientific approach to evolution. I couldn't put it down. So it was a real blessing and a privilege to have you on today to discuss that topic. I think it's really important, and I'm really excited that our audience here got to hear all this incredible information. So everybody in the audience, share this video. Please get this out there to as many people as possible. It is important that they hear these things being discussed. So I want to hand it over to you, Dr. Stadler, for any final words, any final thoughts, and also thank you again so much. I really enjoyed this.
Great. Just thank you, guys. I really appreciate you having me on the show here and the opportunity to share, and just tell everyone to get out your drinking straw and remember this when you're reading the newspaper or looking online tomorrow.
I love it. That's awesome. And Matt, thanks again for joining as helping with this interview.
Yeah, no problem. Anytime. It's great.
Okay, guys. Well, I'm glad everybody enjoyed. This 90 minutes has flown by. So Standing for Truth is out, and everybody, God bless.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.